please.
Hello everyone, my name is Marcin Mizianty and I would like to present sequence-based prediction of protein crystallization, purification, production propensity. The algorithm I designed with Professor Łukasz Kurgan. And let me just start with a fast outline of the presentation. So first I will start with a short introduction to the topic, and then I will follow with the layout of the algorithm architecture, and then I will show how good is our method. Then I will summarize the key points and present you web server, and at the end I will handle your questions. So as we all know, the main method for the prediction of solving tertiary protein structures is X-ray crystallography. More than 85% of the PDB protein structures are solved by this method. But unfortunately, this is quite expensive method, and only 2 to 10% of pursued targets are actually yield high-resolution crystals, which means that more than 60% of the cost is consumed by failed attempts. To elevate that, from around 2004, the information about both successful and failures are stored to help the development of a method which can actually predict the outcome of crystallization. So actually the first thing the first database was the Presage database in 1999, but by that time only the information about the solved crystal structures were stored. The Target DB extended this database, but still only the successful attempts were stored. And in 2004, protein expression, purification, and crystallization database was created. And at that point, the computer scientists, the bioinformatics, had all the data they needed to start developing algorithms to predict the outcome of crystallization. So starting from 2006 up to this year, 10 methods were already discovered. So this pretty new field. What's the motivation? As always, is the money. So the production of high-quality crystals is a major bottleneck. But actually, the structural genomics centers have a certain flexibility in target selection. So they can choose different targets. And if there is a method which could predict the crystallization outcome, this could guide the researchers in selecting targets, so they can select targets which are more likely to crystallize and save some money, not pursuing the targets which are really hard. So what's new in our method? It's actually the biggest novelty in this method is the dataset which was used to develop it. So we developed a new protocol to annotate each step of crystallization, and we use the recent data and the large dataset. So the previous methods actually made the prediction of the crystallization outcome. It was binary prediction, whether the protein will crystallize or not. In our approach, we aim at each major step of crystallization protocols and predicting propensities for each step. We also developed some new features which are really helpful to investigate the crystallization propensity. So the 4 major steps which I was talking about are production of protein material, which is sequencing, cloning, and expression. Then there is a purification step and crystallization, which actually means that the protein crystals were created, and also they were high quality, so the diffraction results were really good. And we also have, in our approach, we are predicting each of these steps, so whether or not the crystallization will pass the step, and we also predicting if the structure was successfully solved. So this is the layout of our algorithm. We're using the input sequence and the energy and hydrophobicity-based indices, as well as the predicted solvent accessibility by real spine, predicted disorder by DisoPred2, and predicted secondary structure by PSI-Pred. Then we generate the features. We're using 10 up to 14 features for each the crystallization step, and we're using SVMs for each step to predict the propensity. Then we aggregate all of these propensities to produce the 4 class labels, like this material production failed, purification failed, crystallization failed, which means that the protein was actually not crystallized, or diffraction quality crystallization successful if we predict that the protein will be crystallized. So I mentioned that we aggregate the propensities from each step. We are using order-based aggregation of SVMs. We're doing it by following the steps of crystallization protocols. So we're starting with material production, and we're checking if the propensity of corresponding SVM is above threshold. If it is, we're selecting this step as our final prediction. If it's not, we're advancing to the next step and checking the propensity. If none of the SVMs produce probability high enough above the threshold, we're selecting the step with the maximal probability as our final prediction. So here are the features which we selected. As you can see, we selected different sets of features for each step, and most of them are based on hydrophobicity and energy-based indices, as well as composition of amino acids, and also predicted solvent accessibility plays an important role. Most of these features will not be of any surprise to the crystallographers, because they already know that these protein characteristics are important when predicting when doing crystallization. But what we've done, we merge, for example, the amino acid with information of solvent accessibility. So we found out, for example, that the content of bird serine is more important than just the simple composition of the serine. Also, for the isoelectric point, it's the common feature in this field. Almost all predictors are using it. But what we found, it's actually, in our experiment, it was actually connected only to purification step. So we also show up which features can be assigned to which steps. We evaluated our method on the independent test dataset. We reduced sequence similarity, but we reduced it only within classes. That means that we didn't reduce, if we have, for example, protein which couldn't produce any material, and we have a really similar protein which actually was crystallized, we didn't remove any of them, because it's shown that even the slight changes in the sequence can change completely the output of crystallization. This makes the prediction harder, because we have plenty of proteins which are similar to each other, but they have different class labels. but this models reality in the better way. We use the standard quality measures like accuracy, Matthews correlation coefficient, area under ROC curve, specificity, and sensitivity. We develop one data set with 4 class labels, and then to train the SVMs, we develop 4 data sets with 2 class labels. We have the data set with to train the SVM for material production step. So we take as negative examples all the proteins which did not produce material, and the remaining as positive. For the purification and crystallization, we didn't take the proteins which did not pass the production of material failed, and for crystallization we also didn't include the purification, because we don't really know if the protein was able to advance through these steps. Would it be easy to purify them and crystallize? It's like I said, even the slight changes in the protein could help, could make it pass the whole step. So we didn't want to include these false, these samples in our method, just to make it predict better. But at the end, we making the prediction on the whole data set. So we checking all the examples. And here, for the 2-class prediction for the crystallization output, we can actually compare our method against the older approaches. And as you can see, the older approaches has the MCC between 0.1 to 0.21 and accuracy between 47 and 56, with except of Ixtal-Pret, which actually achieve MCC of 0.28 and accuracy of 64%. But our method greatly improve over these predictions and achieve MCC of 0.47 and accuracy of 76.8%. We checked with statistical significance test that our methods, that our results are better, statistically better. What you can see here is that actually our method has high specificity, so it tells you exactly with which proteins do not crystallize. For the 4-class predictions, we couldn't compare with previous approaches because, as I told you, they were designed to predict only between if the protein could crystallize or not. So we developed the blast-based approach, which is quite simple. It just looks for the closest homologue in the training data set and takes this homologue label as the prediction. We also compared with the SVM used to predict the given step. What is interesting that these aggregated results are better in terms of MCC for each step, even better than the SVM, which was developed to predict the predictions, to predict this step. It's almost also the case with accuracy, except for the purification failed and crystallization failures, where it's slightly lower. We also have the results for the 4-class prediction, where we again compared against blast-based method, and also we try max-based aggregation, where we simply took the label with the highest probability. But again, the order-based aggregation of the results achieved the highest mean MCC and accuracy at the level of 0.35 and 55.6%. You may actually wonder why we have so much better results than the previous approach. As the time goes, the crystallization protocols changes, they update. So we have more knowledge now. So the older methods were developed on the data sets which takes the proteins from 2006, 2007. And here I present two graphs which shows how the quality of methods drops as the times go. As you can see, they were slightly higher in 2006 for the data from 2006, but when we took the data from 2009, they dropped to the 0.1 in terms of MCC. This is true for all the older methods except XTAL-PRET, and XTAL-PRET was actually built from the newer dataset from 2007, 2008, and it uses homology to PDB, which was updated, I believe, at the end of 2008. So it has some newer data, and this is why it's important to develop new methods which can actually respond to the current protocols. So just to summarize, we designed the improved protocol to annotate the crystallization steps, and based on this protocol, we designed Dataset, which we use to create the novel predictor called PPC-PRET. This predicts the success and failure for 4 major steps in crystallization protocols. It significantly outperforms the several existing predictors and achieves the accuracy at 78.6 and MCC at 0.46 for the prediction of crystallization output. And for prediction of the 4 major steps, the accuracy is about 56%. You may think it's not really high, but given that only 2 to 10% of the pursued targets yield high quality crystals, we believe that the researchers can use output of our method to guide in the target selection. Also, since PPC-PRET identifies the step which halts the crystallization, crystallographers can actually modify their target, include some mutations to help it advance through that step. We also show and confirm several interesting markers that influence the crystallization and all of these mentioned steps. Before I show you the web server, I would like to thank Dr. Helen Berman and Dr. John Westbrook, the curators of PPCDB and TargetDB, for the help in handling the databases, designing of the annotation protocol, and for their constructive comments on the draft of this paper. And we also would like to thank the Natural Science and Engineering Research Council of Canada for funding Professor Kurgan, and Isaac Walton Gillen Memorial Scholarship for funding me. We would also thank the ISCB for granting the travel fellowship. Below you have links to our group page and for my personal website. And the web server is available at this address. It's pretty simple. You just provide the protein sequences your email address, and when you press start, it will generate the results for each protein sequence you entered. It will show it will show at which step the crystallization will stop. It will show you that actually it will crystallize. It also shows the propensities for each step. And if you have any questions, I'm happy to answer them right now.